Hey guys, so today what we're going to do is we're going to finish up talking about chapter 23, and in this case what we're going to be talking about is absorption of our nutrients. Okay, so what we're going to be drawing is absorption process. So again, this is going to be taking place in the small intestines and is following our chemical digestion of our nutrients that we've basically consumed via our diet. And so before we jump into the absorption process, let's draw a few structures. So we're going to draw our blood vessel. And again, don't forget our blood vessel is the ultimate goal, is to get our food into, or our nutrients into our blood vessel so it can be transported to the cells of the body to help us create energy. We also have our epithelium, okay? And again, this epithelium is the epithelium of the small intestine, because this is where the majority of our nutrients are going to be absorbed. So this is our epithelium here. Okay, and then up here, this is where the lumen of the small intestines is. Okay, so again, we've done all of our chemical digestion, and so now what we have are these basic building blocks that we're going to absorb through our epithelium and into our bloodstream. Now, absorption is going to occur via different mechanisms based on the nutrient that we are trying to absorb. So the first nutrient we're going to talk about are our monosaccharides. So we have our monosaccharides, and in this case, our three monosaccharides, major monosaccharides that we're going to talk about are glucose, galactose, and fructose. Okay. Now, glucose and galactose have a different mechanism of transport into the epithelium than fructose does. So, fructose is a fairly easy one to kind of remember, and that is because fructose is actually going to enter the cell via a facilitated diffusion process. So I always draw my facilitated diffusion for some reason as a little triangle. Okay, so this is going to enter the cell via facilitated diffusion. And again, remember, facilitated diffusion, all that means is that we need a carrier molecule, a protein, in that plasma membrane in order to serve as a slide for that fructose molecule to get through. Okay, then, so that's on the apical surface, and then on the basolateral surface, this is going to leave the epithelium through the apical surface, again, through a facilitated diffusion mechanism, okay, and enter into the bloodstream. Okay, so that's strictly fructose. Okay, now glucose and galactose move via a little bit different mechanism. And before we can actually talk about what happens on the apical surface, first we need to talk a little bit about what's happening actually on the basolateral surface. So on the basolateral surface of this epithelium, what we have is we have a sodium potassium, whoops, let me go back. You guys can actually read that. Okay, our sodium potassium pump. And if you remember our sodium potassium pump, this requires ATP, okay? And the reason it requires ATP is because what we're going to be doing is we are going to be pumping sodium out of the cell into the surrounding environment, okay? Where we know from AMP1 that the most abundant ion, or that sodium is more abundant outside the cell. And then what it's going to be doing is pumping potassium into the cell. And we know that the abundance of potassium inside the cell is much greater than outside. So we're pumping against its concentration gradient, its natural concentration gradient. And therefore this requires energy in order to pump the sodium and the potassium into the corresponding environments. Okay, so since this requires energy, this is what we would call a primary active transport system, because it uses ATP directly. Now, as a result of this pump, Okay, so as a result of this pump, sodium levels inside the cell become low. Okay, so as a consequence of this pump, well, I don't know how to do this. So the pump causes a decrease of sodium in cell. Okay, does that make sense, right? So if we're removing sodium from the cell and pumping it out, that means we now have a deficiency of sodium inside the cell. So if given the opportunity, if sodium resides out in this lumen, okay, and we provide a mechanism for the sodium to move, it's going to want to move into the cell. And we actually have a mechanism that exists on the surface of this cell. And this mechanism is what we call a co-transporter. And I'm just going to abbreviate it CT. Okay. So what this co-transporter does is this co-transporter allows sodium to enter the cell. Okay. And what happens is in the sodium moving into the cell, our galactose and our glucose can jump on this co-transporter with the sodium and enter the cell. Now, this is not a facilitated diffusion process, even though we're using a co-transporter. And it's not a facilitated diffusion process because the fact that we're creating this concentration gradient deficiency, okay, via an ATP process. So this is this is what's called a secondary active transport mechanism. Okay, so in order for that glucose and galactose to get into that cell, it requires the ATP pump, the sodium potassium pump, to be working. Without that sodium potassium pump, you don't create the deficiency of sodium in the cell. You have no reason for sodium to enter the cell via the co-transporter. If sodium doesn't enter the cell via the co-transporter, you can't get galactose and glucose into the cell. Okay? However, once into the cell, on the apical surface, okay, glucose and galactose leave the basolateral surface via facilitated diffusion into the blood system. Okay, so again, this is how our monosaccharides are transported through our cells. Okay, now amino acids. Okay, so again, if we look at our breakdown product of our proteins, okay, what we end up getting are amino acids. Okay, which is our kind of the second thing we're going to talk about. And again, these amino acids, we have 20 different types of amino acids. And amino acids are actually going to enter the apical surface via a co-transporter. Okay, so we have another co-transporter here. So our sodium is going to enter via our co-transporter. And then what's going to happen is our amino acids will jump on and leave or enter the cell via, again, this is a secondary active transport. And again, I didn't draw here, but again, this relies on that sodium potassium pump we talked about when we talked about the absorption of our monosaccharides, specifically our glucose and our galactose. And then again, just like in our monosaccharides, this is going to leave the basolateral surface via, or this in terms of amino acids, via facilitated diffusion. Okay, so again, if you understand this initial process of the monosaccharides, the sodium potassium pump, then understanding the amino acids becomes a little bit more self-explanatory, or a little bit more easier to understand. Okay, we have next our nucleic acids. Okay, and our nucleic acids again, are the breakdown product of our, well, so actually, are they really nucleic acids? At this point, they're not. I'm going to go back and erase this. Okay, because actually what we're talking about are nucleotides. So our nucleotides, which are the breakdown product of nucleic acids, are absorbed a little bit differently in that they're absorbed through the apical surface via active transport. Okay, so they don't require a co-transporter. Instead, they're going to require just an ATP process. So this is going to be primary active transport. Okay, so these enter then through primary active transport. This goes down and actually leaves the basolateral surface also through a primary active transport. Okay, so again, monosaccharides, amino acids, going to enter the apical surface via either a secondary active transport or facilitated diffusion if you're talking about fructose. Nucleotides are going to enter through a co-transporter system. I'm not sorry, through an active transport system, so a primary active transport system. Okay. And I'm just going to kind of scroll us down. I'm going to redraw our blood vessel down here. Okay. I'll redraw our epithelium because we have a few other things that we need to cover or finish covering. Okay. Okay. So, again, here's our epithelium. Our lumen's up here. Okay. And so, one other thing we have is our lipids. Okay. And our lipids, our lipids are a little bit unique. And again, when we're talking about our lipids, oh, let me go back. We're actually not talking about our lipids. Because what are we really talking about? Okay. What we're actually talking about are our fatty acids and our monoglyceride. Okay, which again is our breakdown product of our lipid digestion. And so what we end up having is we end up having in the lumen these little droplets of fatty acids and monoglycerides, okay, that have actually been surrounded by, you guys remember what these little droplets are surrounded by? So these are surrounded by bile salts. Okay, so these are bile salts. And again, remember our bile salts are emulsifiers, so it creates these little microscopic droplets, which allows for the lipids to be broken down into these fatty acids and monoglycerides. And so what ends up happening is these bile salts end up coming down and fusing with the membrane and allows for our monoglycerides and our fatty acids to enter the cell. Now, if we go back to our AMP1, we know that because fatty acids and monoglycerides are lipids, they enter via simple diffusion. Okay, and they enter via simple diffusion because they're lipids, and we know that the phospholipid bilayer allows lipid-soluble things to enter through it. Okay, so this process happens via a simple diffusion process. Okay, now where this becomes a little bit different than our other nutrients we've talked about is that these fatty acids, so now inside this cell we have our fatty acids and our monoglycerides. Okay, these actually get sent to the smooth ER. So these get sent to the smooth ER, and I'm just going to kind of draw the smooth ER like this. Okay, so the smooth ER. Okay, And in this smoothie are these fatty acids and monoglycerides are converted, okay, into triglycerides. Okay, and again, remember, triglyceride is just a type of lipid that exists in our body that we can utilize for energy. These triglycerides are packaged in a complex known as a chylomicron. This is sent to the Golgi apparatus. Okay, the Golgi then packages, okay, these chylochromes. Okay, and these chylochromes are then exported via exocytosis, okay, not into the bloodstream, okay, but into the lymphatic system. Okay, so these then enter the lymphatic system, more specifically the lacteals, or via the lacteals. Okay, let me go back and actually, so you guys can actually read what I wrote here. So lymphatic system, the lacteals, okay, which is just a portion of the lymphatic system. Okay, these then go out and get stored within the cells themselves. Okay, now the last few things that we didn't talk about yet are our vitamins as well as our water. So water is really easy. Water is going to enter, so water, okay, 95% of it will be absorbed in the small intestine, and it's going to be absorbed via osmosis. Okay, and our last component we're going to talk about is our vitamins. Okay, and the vitamins, depending on what they are, will be absorbed via various processes. So we have kind of our dietary vitamins. We have vitamin C and B. Okay, this is made by bacteria. We have our vitamin D, E, A, and K. Okay, and then we have vitamin B12. Okay, and so these all take a little bit different process. Our dietary vitamins and our vitamin B and C, okay, are going to enter via diffusion, okay, or transporters. Okay, again, that's our that group. Okay, our vitamins D, E, A, and K, these are fat soluble and actually become part of the micelle that's created over here in the absorption of fatty acids. So these are going to enter via passive diffusion. Okay, and then our last one is vitamin B12, and vitamin B12 is a little bit unique because vitamin B12 has to bind to intrinsic factor in the stomach. Okay, and then this binds to a receptor and enters the cell via endocytosis. Okay, so Again, this is basically how we carry out our absorption. Again, we have our vitamins, our water, our fatty acids, and our monoglycerides, as well as our nucleotides, our amino acids, and our monosaccharides. So once into the body, okay, these nutrients will be then utilized via the cells and creating we'll either store it or we'll use it to create energy and ATP. And we'll talk about that process of nutrition metabolism when we get into chapter 24. Any questions? Make sure you guys shoot me an email or bring your questions to class. I'd be happy to answer any of them.